This lecture block of lifespan developmental psychology is entitled Biological and Environmental Foundations. A genotype is a person's genetic makeup. A phenotype is a person's observable traits, what can actually be seen. If you have curly hair plus you have a gene for straight hair, your phenotype is curly hair. Your genotype includes both. The basic unit of heredity or biology is the gene. Genes are always influenced by the environment and by the person's developmental history. From your parents, you inherit genes, also called segments of DNA, located on chromosomes. Chromosomes transmit and store genetic information. They are rod-shaped structures comprised primarily of DNA. Every cell, except reproductive cells of eggs and sperm, have 23 pairs or homologs of chromosomes, or 46 chromosomes in all. Each homolog has a pair of chromosomes, one of which was contributed by the mother, and one contributed by the father. DNA, deoxyribonucleic acid, is a chemical substance that can duplicate itself via mitosis. It is a twisting, long molecule in the shape of a double strand connected like in a ladder. Each rung of this ladder has two base chemicals that join to the sides of the ladder. There are 4 base chemicals, and adenine always pairs with thymine. Cytosine always pairs with quinine. A pairs with T. C pairs with G. Genes are different lengths. Some 100 rungs, and some several thousand rungs. Each cell has 30,000 genes on the chromosomes. The majority of these genes are identical for every human and are called monomorphic genes. Some of them are called polymorphic genes and establish individual uniqueness. Genes do their jobs by sending recipes for making proteins into the cell's cytoplasm. Once made, proteins trigger chemical reactions. These crucial proteins number 10 to 20 million different varieties, and they are modified by environmental factors. So it is not just biology operating as the genes send out their instructions and recipes. Even at the cellular level, there is an interaction of biology and environment, nature and nurture. Mitosis is cell division that duplicates chromosomes before dividing into daughter cells. Every cell in your body, with the exception of gametes or reproductive cells, reproduce by mitosis. In each new cell, you end up with 46 chromosomes, identical to those chromosomes in the original zygote. Reproductive cells or gametes, sperm and eggs, reproduce by meiosis. Meiosis is cell division which results in the sperm or egg being reduced to 23 single chromosomes instead of 46. When an egg is fertilized by a sperm, the resulting zygote will have the 46 chromosomes needed for normal development. During this cell division of meiosis, random assortment of genetic material is created by means of crossing over. Chromosomes pair together and exchange segments of DNA or genes. This mixes things up and provides needed variability. After meiosis, there are 4 sperm cells or male gametes. Meiosis continues throughout the lifetime of the man, producing new male gametes or sperm and allowing him to father children throughout his lifespan. After meiosis in the female, only one ovum results. When born, the female has all of her ova in her ovaries, 350 to 450 of which will mature during her lifetime. She can bear children for 30 years or so. Of the 23 pairs of chromosomes, 22 pairs are called autosomes, and the 23rd pair are sex chromosomes, either two X's for a female or an X and a Y for a male. The mother donates an X, and the father donates either another X or a Y. Conception takes place when the male sex cell, or gamete, called a sperm cell, joins or fertilizes the female sex cell, or gamete, called an ovum. The result is a single cell called a zygote. The ovum is the largest cell in the body, 1/4 the size of a period at the end of a sentence. A girl is born with all 400,000 of her ova on board. An ovum lives only 24 hours after release. The sperm is one of the smallest cells in the body, only 1/600th of an inch from head to tail. Sperm are made by the testes of a mature male to the tune of several 100 million each day. Sperm live up to 4 days inside a woman's reproductive tract. Fertilization is most likely to happen if intercourse happens during the 5 days before ovulation and the day of ovulation itself. If there is no conception, the sperm are absorbed by white blood cells, and the ovum exits via the uterus and vagina. 1/3 of twins result from a single fertilized ovum that splits into two, and are called monozygotic twins. These twins are the same sex and have the same DNA, but they are not identical in temperament and can have mirror image characteristics, like one is right-handed and one is left-handed. They can have hair or dental patterns that are on opposite sides. One can be born with a cleft palate and the other with a complete fusion. Though the twins are identical, they do not have identical prenatal and postnatal experiences. 2/3 of twins result from different sperm cells, and usually 2 ova, and are called dizygotic twins. They are no more alike than any 2 siblings. For the first 6 weeks, the primitive gonad tissues look identical in males and in females. Sexual differentiation begins around week 7. For the first 7 weeks following conception, the developing brain is feminine. Things do not change unless there is a Y chromosome present to trigger greater quantities of testosterone and other hormones associated with maleness. In the 7th week, the Y chromosome triggers differentiation into testes. If there is no Y chromosome present, then the gonad tissues begin differentiating into ovaries. After this, further sexual differentiation is controlled by self-produced hormones. Steady, high levels of testosterone are needed for male sexual characteristics. Even an XY genetic male will develop a female body plan if not exposed to high male hormonal levels. Prenatal hormones do more than affect the development of genitals or primary sex characteristics. They also affect the hypothalamus, determining whether it will be male differentiated or female differentiated. The hypothalamus controls the pituitary gland, so it is a major player in the production and release of sex hormones. Prenatally, an individual is programmed to behave in certain ways, like sexual behavior. But it is not until this behavior is activated at puberty by certain changes in hormone levels that the behavior can take place. A person has the brain organization that determines sexual behavior, but does not have the motivation to engage in this sexual behavior until after the hormonal changes of adolescent puberty. Alternate forms of genes for the same trait are called alleles. The alleles for blood type are A, B, and O. Blood type gene is found on chromosome number 9. Some alleles are dominant, some are recessive. Dominant ones for blood type are A and B, and they mask the presence of recessive ones, which is an O for blood type. 6 possible genotypes exist for blood type: AA, AO, BB, BO, AB, and OO. Only 4 phenotypes exist: A, B, AB, and O, because the recessive O gene can be masked by a dominant A or B. Other physical characteristics following a dominant pattern are curly hair, dark hair, dimples in the chin or face, far-sightedness, Rh-positive blood, double-jointedness, type A blood, type B blood. Physical characteristics that follow a recessive pattern are straight hair, blonde hair, red hair, patterned baldness, nearsightedness, sickle cell anemia, Rh-negative blood, type O blood, albinism, some forms of deafness. Please note here that most physical characteristics are governed by multiple genes called polygenic inheritance. Eye color is one of these. It is possible for 2 blue-eyed parents to have a brown-eyed child, even though brown is usually dominant. If only 2 genes were responsible for brown dominant and blue recessive, then it would not be possible for 2 blue-eyed parents to have a brown-eyed child, since they would not have a gene for brown in their genotype to donate. Skin color is controlled by 3 or more sets of genes on 3 different chromosomes, so it is another example of polygenic inheritance. As for genetic defects, 90 to 95% of all conceptions involving chromosomal abnormalities end in miscarriage, many only days after conception, before the mother even knows she is pregnant. Only 31% of all conceptions survive prenatal development. 69% do not survive it. Genetic and chromosomal abnormalities are not always evident at birth. Tay-Sachs disease is a central nervous system degeneration. It's most commonly found among Jews of East European ancestry. Tay-Sachs involves an error in metabolism. There is an increased amount of glycolipid in cells of the nervous system, causing them to swell, rupture, and die. There is decreased motor ability, increased mental retardation, and death by age 3 years. Tay-Sachs shows up after 6 months of age, as does sickle cell anemia. Sickle cell anemia is a blood disorder most common among African Americans. Deformed, fragile red blood cells in the shape of a sickle or crescent instead of being round. These fragile red blood cells clog vessels, depleting them of oxygen. This blocks the flow of blood, causing intense pain and swelling. Both Tay-Sachs and sickle cell anemia are due to recessive inheritance and need genes from both parents to affect the child. Sickle cell anemia is said to have incomplete dominance because a person with only one recessive gene will demonstrate mild temporary symptoms under the stress of oxygen deprivation at high altitudes or after strenuous exercise. 85% of victims live to adulthood. The average life expectancy is 55 years. Cystic fibrosis is characterized by too much mucus, causing lung and digestive problems. It may wait as long as age 4 to show up. Mucus obstructions are especially found in lungs and digestive organs due to a missing enzyme. Most victims live to middle adulthood. Recessive gene and spontaneous mutations are responsible, and research is ongoing, and the future is even brighter now. PKU is another single gene dominant recessive disorder. It's called phenylketonuria, PKU. It's a metabolic disorder caused by the presence of two recessive genes. 1 out of 8,000 births are afflicted with PKU. It can be screened for after birth with a PKU heel stick test. In order for a baby to have the symptoms of PKU, the baby must also have the environmental agent of phenylalanine, an amino acid. PKU involves an inability to break down phenylalanine into tyrosine, and it causes mental retardation and brain damage. But if you alter the diet by age 6 weeks, you can cure PKU. You do not give the baby milk. Milk has phenylalanine in it. This amino acid is found there, and it will build up, and the resultant brain damage will happen. The PKU recessive gene leads to fair coloring. It affects more than one trait, but again, it can be cured with diet. Sex-linked inheritance refers to recessive disorders linked to the X chromosome that puts affected males at a disadvantage. The Y chromosome is only 1/3 as long as the X chromosome, so it lacks the corresponding genes to override a harmful recessive allele on the X. Another X chromosome has more chance to have a dominant allele to counteract the recessive one. Red-green color blindness and hemophilia are examples of sex-linked inheritance. Genetic imprinting is a phenomenon that chemically marks one allele to become activated regardless. Children with a diabetic father are more likely to develop diabetes than if their mother has it. Children with a mom with asthma or hay fever are more likely to have one of these than if their father suffers with them. Mutation is how harmful genes are formed in the first place. It is a sudden change in a portion of DNA caused by chance or by teratogens, which are harmful environmental agents. A mutation may affect only 1 or 2 genes or many more. High-energy ionizing radiation, at this kind of exposure in the mother or the father, can result in germ-line mutations. This damages the cells, which will become gametes after meiosis. The damaged DNA is passed on. Somatic mutations happen with normal cells, which mutate at some time during life, with probabilities increasing with age. After this, each cell derived from the mutated ones become widespread enough to cause disease like cancers, epilepsy, and heart disease. Serious developmental problems are most often caused by chromosomal abnormalities which occur during meiosis, when chromosomes do not separate correctly or parts of one translocate or break off. Down syndrome is the most common chromosomal abnormality. It occurs in 1 out of every 800 live births. In 95% of cases, there are 3 copies instead of 2 for chromosome number 21, and the condition, Down syndrome, is called trisomy 21. Part of chromosome 21 can be translocated onto another chromosome. 97% of cases are due to chance abnormalities during meiosis, the production of gametes. It's usually due to abnormalities in the ovum, but 5 to 10% of the time, the extra chromosome is donated by the father. A mosaic pattern of Down syndrome may occur if there is an error in the early stages of mitosis, causing mutations in some, but not all, cells. Less genetic material is involved with the mosaic pattern, and the symptoms are not as severe. The greatest percentage of Down syndrome victims are mentally retarded with speech and memory problems. Physical features include a short, stocky build, a protruding tongue, almond-shaped eyes, and an unusual palm crease. A few have normal or above average intelligence and can learn well. 75% of them are miscarried. Easygoing, delightful youngsters are those with Down syndrome. If you've ever known a child with Down syndrome, you know what I mean. These people do well in structured jobs. More than 70% live into their 60s. There is a high risk for Alzheimer's. There's a high risk for heart and intestinal defects and leukemia in childhood. Those with Down syndrome are often born with eye cataracts. One of a set of identical twins can have Down syndrome, and the other of the set of twins be normal. The risk of Down syndrome is greater with older parents. In the case of the mother, the ova has been around a long time, since before birth, and may weaken with time and with exposure to teratogens. Sex chromosome abnormalities refer to problems with number 23 pair of chromosomes. 1 out of 400 babies are born with sex chromosome number 23 anomalies. XXY is Klinefelter's syndrome. There are 2 X's and 1 Y. The child is a boy. Boys, for the most part, look normal with long legs and arms and underdeveloped testes. They do not usually have mental retardation, but they do often have language and learning disabilities. Children with Klinefelters are infertile when they grow up. Secondary sex characteristics, like the voice and the penis, do not develop normally. Possible breast development during puberty and high-pitched voice and light beard growth are experienced. XYY is rarer. This is 2 Y's and 1 X. There's no name that goes along with this one. Victims are unusually tall with low IQ, and they're prone to acne. XO is called Turner's syndrome. There is only 1 X chromosome. 1 out of every 3,000 girls are born with Turner syndrome. They experience stunted growth, short, immature look, a webbed neck, eyelid fold, receding chin, a broad chest, and they are sterile. They have normal and above normal IQ. Their verbal abilities are normal or above average, but they have poor spatial abilities and poor recognition of facial expressions of emotion. Secondary sex characteristics do not develop, like menstruation and breast, without supplemental estrogen therapy. XXX, called trisomy X, have victims with mental retardation, but who look normal. They also have poor verbal abilities. Fragile X syndrome only occurs when the damaged X chromosome is donated by the mother rather than the father. There is an abnormal repetition of the bases or rungs of a DNA sequence. This gene defect results in mental retardation, but is only expressed when passed from mother to child. Fragile X syndrome affects 1 out of 1,200 males, 1 out of 2,000 females. It's inherited. Victims have a long, narrow face and prominent chin and ears. They are extremely shy with inadequate social skills. It is the most common inherited cause of MR, mental retardation. In this lecture block, much emphasis has been placed on DNA, chromosomes, and genes. But please remember reciprocal determinism, the two-way interaction between the organism and his or her environment. René Spitz in the 1940s found that early nurturing and stimulation are essential. He studied 2 groups of at-risk babies. One group was in an orphanage, one in a nursery with contact with prisoner mothers. Both institutions were similar. They were clean, with adequate food, clothing, and medical care. The prison moms fed, nursed, and cared for their own babies, though the hours were limited. Normal development was found. Orphanage babies had 1 nurse for 8 babies. Except for brief feedings and diaper changes, babies were isolated in cribs with sheets over the sides to prevent the spread of infection. There was little or no stimulation, nothing to look at or play with, and a bare minimum of human contact and affection. Many of these babies didn't make it to their 2nd birthday. Those who lived 2 years were stunted physically, cognitively, and emotionally, and they were highly susceptible to infection. Many were severely retarded. By age 3, most of the survivors couldn't walk or talk. They were apathetic and withdrawn. In contrast, the prison nursery babies were normal and exuberant. Genes are important for healthy development of nerve cells, but so is experience. This classic study brought it home so emphatically. Neurons, nerve cells, are remarkably plastic. Brain tissue is molded by experience, sight, sounds, thoughts. Development is not fixed, but dynamic and constantly updating. Each experience imprints neural circuits and modifies how future sights, sounds, and thoughts will be registered. Brain development is a delicate dance between nature and nurture, between genes and environment. Genes program the sequence of brain development. The quality of brain development is programmed by the environment. Genes are like gravity pulling downward. Environment is the rocks, the trees, the holes which divert that pull of gravity in a specific way. A recommended book that touches on a lot of this is called What's Going On in There by Dr. Lise Elliott. It was published in '99. Now we are going to follow the environmental example of poverty and show the complex interaction between social economic factors, parents' psychological well-being, and the socio-emotional functioning of their children. We are going to see the interplay that Bronfenbrenner says exists among contexts, settings, and how these interactions shape and influence the development of children. Let's begin from the outermost circle of Bronfenbrenner's diagram of context and work our way into the center. The issue we are tracing is poverty, and particularly poverty among inner-city families. The cultural context. The culture that we are examining here is that of the United States. U.S. culture stresses independence and individuality and proclaims that success is the result of individual effort. People who fail have only themselves to blame. For families struggling in poverty, this only adds insult to injury. This only adds psychological insult to financial hardship. For minority families struggling in poverty, there are the added social and economic barriers presented by cultural attitudes about race. The social and economic context. Chronic poverty is increasing for inner-city families because of several factors. Cities are moving away from traditional working-class jobs and transforming from manufacturing centers to information processing centers requiring more technological training. Jobs that are available to inner-city people with low levels of education also have low wages. Households headed by women with little income and resources are increasing. The immediate context. Poverty restricts the freedom of choice in most arenas of life, such as choice of neighborhood, housing, quality and level of schooling, recreational opportunities, and health care. Poverty also subjects adults to the control by other people, such as social workers, and subjects children to less positive attention and encouragement, and more criticism and punishment by teachers and school officials. Children are viewed as problems ready to happen. Poverty subjects families to a string of small and large crises with little time to recuperate before the next one hits. There's inadequate housing in rundown neighborhoods, exposure to daily danger from rats to bullets, depressed parents with constant stress, anxiety, and psychosomatic problems, poor health, alcohol, drug abuse. Poverty undermines existing marital relationships and reduces the possibility that couples will marry in the first place. Poor inner-city households are increasingly being headed by women, while males are increasingly becoming unemployed or earning wages below the poverty level. All of these contexts, cultural, social, economic, and immediate, negatively affect the parent's ability or willingness to be supportive and consistent and involved with their children. Keep them in line is the main thrust of child rearing. Fathers, in particular, feeling powerless in the outside world, find a need to wield their power in the family, asserting themselves harshly and arbitrarily. Child neglect and abuse increase under these circumstances. For inner-city poor families with strong extended family ties and community involvement, the picture is much brighter. The burden of child rearing is shared and distributed. Children of single mothers who live with their mothers and or grandmothers experience a higher quality of parenting skills, provided the older generations have these higher quality skills of parenting themselves. The biological context. The quality of parenting a child receives depends in part on the child. Children born to impoverished inner-city families are far more likely to be born to young, malnourished mothers who received little prenatal care and who used drugs and or alcohol during the pregnancy. This results in babies who are more likely to be born prematurely, or more likely to be difficult babies, or more likely to need extra care and attention from mothers less able to give it. These babies are also more likely to be sick more often, injured, or malnourished, leading to additional developmental problems. Those babies receiving intervention, including psychosocial stimulation, have been found to improve.